The second speaker is Dr. Ritu Aneja from, she's a distinguished professor at the Georgia State University, Atlanta, and I'd like to inform the audience that she actually did a PhD in chemistry from Delhi University. She is also the director of Molecular Basis of Disease program at the same university. She serves in several journals as handling editor or member of the editorial advisory board, including Scientific Reports and World Journal of Experimental Medicine. She's going to talk to probably the hottest topic in breast cancer research on triple negative breast cancer landscape, research resources and global initiatives. Dr. Ritu Aneja, sorry.
Okay, so let's begin with a thought exercise. So let's all imagine we are in the United States. And you have 10 of your female friends and relatives in this room with you today. One of them is going to get breast cancer. And if she is a white woman, then 1 of every 10 with breast cancer will have the triple negative subtype, which is one of the most aggressive breast cancer subtype. And if she happens to be a black woman, then 3 of every 10 with breast cancer will have the triple negative subtype, and each one is twice as likely to die from it. Let's come back to India, and let's visualize you have 10 of your female friends and relatives in this room with you. Unfortunately, 3 of them will get breast cancer, and 3 of every 10 with breast cancer will get the triple negative subtype. So here's a quote from Viktor Frankl. He says, between stimulus and response, there is a space. In that space is our power to choose our response. And in our response lies our growth and our freedom. And the response we choose to make is to make a difference in this world by reducing this racial and biogeographic disparity of this deadly monster called triple negative breast cancer. Thank you so much for having me here today. It's an absolute honor. So when I started working on breast cancer about 15 years ago, I was looking at the cell cycle for cell biological traits that can serve as surrogates of intratumor heterogeneity, like centrosome amplification, centrosome clustering, and even tumor cell kinetics, and it was more of a relationship between Ki67 and mitotic index. So with an overall intent to glean prognostic and predictive biomarkers, and of course find some druggable targets that based on the cell cycle. And it was then I was very intrigued by the stark disparity between black and white women, and that was the time when I started slowly drifting towards disparity research. And at this time, my lab has a large focus on several aspects, both biological and non-biological, for breast cancer. So here are some of the questions that inspire my research. So why are tumors in triple negative patients inherently more aggressive than breast tumors in others? Why is there a tremendous biogeographic disparity in triple negative across the world? Are there easily measurable differences among triple negatives that can tell us which cancers will be indolent versus aggressive, or which will respond to chemotherapy versus those which will not? Can we measure those differences and thus better optimized 3NBC treatment? So here is the roadmap for my talk today. I will begin with an overview of the breast cancer disparity in the United States and briefly touch upon the biogeographic disparity in the world. And I'll briefly mention the knowledge gaps that led us to the creation of an international consortia to bridge, called ICARD, to bridge the racial disparity gap. I will share with you some surprising research findings from one of our global multi-institutional study that took the subject matter of triple negative a notch up to quadruple negative, and I will touch upon the multifaceted complexity of disparity and how and why a multi-pronged approach is required to get grips on disparity. Lastly, I'll give you a glimpse of the breadth of my current ongoing projects in my lab. So while the incidence of breast cancer between the blacks and whites in the United States is fairly similar, the disparity in mortality rates is growing over these years. As of today, blacks in the United States have 40%, black women in the United States have 40% higher rates of dying from breast cancer. And a closer look at the various subtypes tells us that black women are disproportionately affected by the triple negative subtype, and this is prevalent across all age groups. This slide has got quite some semblance with the Indian scenario, where we see that triple negative is on the rise, and particularly in premenopausal women. So ironically, triple negative is still described by a catch-all phrase that just lumps together all cancers that lack these lack these three receptor tags, ER, PR, and HER2, and it is sadly defined by what it lacks rather than what it has, which is pretty sad, because what it does not have, is not actionable. So triple negative, which is a disease of multiple negatives, is not a single beast. It's very diverse in gene expression, mutational landscape, clinical behavior, response to treatment. It's got a very dismal prognosis, high propensity to relapse within the first 3 to 5 years, no approved biomarkers or targeted therapies. All commercially available assays such as Oncotype DX, MammaPrint, PAM50, risk of reoccurrence, all categorize triple negative as high risk. And there's extensive intra and intra-patient heterogeneity. You know, in the United States, black women have 2 to 3-fold higher chances of getting triple negative, and they they present at a younger age of about 35 years, more resistant, more aggressive, twice the chances to die of triple negative than compared to white women. Intriguingly, the numbers vary drastically across the globe. If in the United States, triple negative is 10 to 15%, but that is regardless of race. If you bring the race in, it's in black women about 25 to 40%. Numbers in Africa much higher, you know, especially in the Western Africa, Sub-Saharan Africa, countries like Nigeria, Ghana, just as Brittany mentioned, and have much higher numbers compared to those countries that have eastern ties, like the South Africa and the Central Africa. And okay, so blacks Americans in the contemporary United States represent genetic admixtures of several ancestral populations. So the question is, the proportion of African ancestry in an individual is self-reported. So no, single institutions, and African Americans are a minority. Only 12% of the US population is black, so they're considered as a minority. So single institution studies and even multi-center partnerships, they're often underpowered to glean biologically significant differences in biomarkers or genetic features, and which is why we founded this consortium 5 years ago called ICARD, International Consortium for Advancing Research on Triple Negative Breast Cancer. ICARD today is a partnership between 35 institutions from 15 different countries across 4 continents, and a network of 20 teaching hospitals from Africa. So ICARD was born in Nigeria because the first meeting was held in Nigeria at OOU, which is in Ogun State, Nigeria. And the second meeting was in Atlanta. It was NIMHD funded, attended by 200 participants with speakers from 11 different countries. So several different groups, as you know, have tried to capture the inherent heterogeneity of triple-negative breast cancer, starting from the legendary paper from Jennifer Peyton-Pohl's lab in 2011, the JCI paper, classifies triple-negative into 7 different subtypes, and then there have emerged several different classification schemes. Jennifer's was followed by Powell Brown's lab from MD Anderson, that's the Bernstein's classification. But the current day consensus categorizes breast cancers, triple-negative cancers, into 4 different subtypes based upon transcriptional profiling: basal-like immune-activated, basal-like immune-suppressed, mesenchymal, and luminal androgen receptor. And as you know, androgen receptor is an emerging target for breast cancers, and it is highly expressed. It's actually more expressed than ER and PR, and about 35 to 40% triple negatives express AR. So we thought it would be a great idea to classify triple negatives based on AR as AR positive triple negatives, which will be the classical triple negatives, and AR negative triple negatives as quadruple negatives. So there are several clinical trials, about 20-25 of them going on, that are using AR antagonists for triple negative, and they are showing promising data, although there is a lot more to understand about AR-driven biology in triple negatives. And this time we thought, what's the role of AR? We asked ourselves a question: what's the role of AR in prognosis of triple negative? And what we found, that we had all kinds of papers. So there was literature where AR is a good prognosis biomarker in triple negatives. There's reports telling us AR is a poor prognosis biomarker. AR status has no impact. And that's when we thought we would like to, given that we had many cohorts of triple negative from various different countries, so we undertook this global multi-institutional study. And it involved 15 different institutions across 6 countries. So we had a British cohort of 239 samples from Nottingham City Hospital, a Norwegian cohort from Stavanger University, a Nigerian cohort of 180 samples from 3 teaching hospitals, an Indian cohort from RGCI, Delhi, and Tata Memorial, and an Irish cohort from Galway, as well as, of course, an American cohort from these hospitals. So it was a total of about 1,500 samples. We stained them centrally, and except the Indian samples, because of the strictest regulations that we cannot receive Indian samples. So they were stained and antibodies were sent here. Other than that, all were stained and scored with various cut points, 1% centrally. So here's what we found. So here's the percentage of AR cases expressing AR. The Nigerian data set showed us that AR is almost absent. So more than 90% of triple negatives turned out to be quadruple negatives from Nigeria. And then this is the percentage of the other countries. So when we zoomed into the US cohort, we found that QNBC was predominantly associated with African Americans. So the survival curves showed us uncovered quite interesting data here and about the prognostic role of AR in adjuvant treated patients. So what we found that loss of AR was poor prognosis. Loss of AR was poor prognosis in the US cohort, whereas loss of AR was good prognosis in the Norwegian, Irish, and Indian cohort. In the UK cohort, loss of AR or presence of AR did not have any significant influence or impact on survival. And as I said, the Nigerian cohort was lopsided because mostly more than 90% were AR negative. So we next did, for multivariate analysis, we divided our data set into two groups. One where TNBC had good prognosis, the other which was the US cohort, whereas the other was where TNBC had poor prognosis, which is the Indian, Norwegian, and the Irish cohort. So what we found that AR status was significant, was important, remained prognostic even after adjusting for age, chemotherapy, and race. So we took a deep dive into the US cohort where we wanted to ask ourselves a question that where does the racial disparity lie, whether it's in TNBCs versus or in QNBCs. What we found to our surprise that racial disparity was significant in QNBCs and was not significant among TNBCs. So we do not know if triple negatives or even have racial disparity. We still need a validation cohort for this, whereas all the other data that I showed until here has been, we have had a validation cohort, a separate validation cohort. So AA race, African American race, is a significant independent predictor of outcomes. So here's when we dived into TCGA to look at what's actionable in quadruple negatives, because if AR is present, then we can go after AR, but then if AR is not present, then what do we have to go after? We do not know. So there's a dire need for actionable targets in quadruple negatives, which happen to be more associated with black population and women of African descent. So this is all TCGA data, and then we did gene set enrichment analysis to look at what's the top enriched gene ontology. We found that was Wnt beta-catenin. These are waterfall plots where we see that Wnt beta-catenin signaling is in overdrive in AR low patients. Here's again some data from TCGA for beta-catenin expression, and here is data from 167 patients that we recently sequenced, RNA-seqed, and they have a great follow-up data as well. So this is, again, same data, same results, and this is staining of 142 cases for nuclear beta-catenin, and we found that nuclear beta-catenin was higher in QNBCs. So again, so this was to, dual reporter assays to look at canonical Wnt targets and seeing the Wnt downstream genes, make MMP7, so it's all in vitro work showing that all of these genes which are Wnt targets are higher in QNBCs versus TNBCs. Again, more genetic manipulations, knockdown of AR in triple negative cell lines and overexpression of AR in quadruple negatives, showing reduced beta-catenin, E-carnitine, increased expression of CD44, cyclin D1 when we knock down AR in triple negative, and same data when we overexpress in quadruple negative. Opposite, that's what we found. Here is doubling time, where we found that quadruple negatives have a faster cycling rate, they have a shorter doubling time. Centrosome amplification, which is a surrogate for intratumor heterogeneity, was higher in quadruple negative versus triple negative. So this is one gene signature, CA20. It's a 20 gene prognostic signature that we published a couple of years ago in 2017. And that quantifies centrosome amplification. That was again higher in triple negative. And SYN25, which is a very established chromosomal instability signature, that was also higher in quadruple negatives. So higher SYN25, higher CA20, higher centrosome amplification, shorter doubling times, Wnt, beta-catenin in overdrive, all indicate aggressive biology in quadruple negative. So coming back to the question as to why AR loss is good prognosis in Indian population, Norwegian, and Irish that we did in our hands is still a mystery. And we have charted out a few steps here. We wish to look at splice variants, risk factors, differences in tumor microenvironments, gene expression studies, sensitivity, and then also looking at molecular apocrine corrector. So all of this is underway. So just wanted to end by saying that aggressive biology is just one of the reasons that underlies racial disparity. There is a lot of other factors. So, as Brittany had alluded to in her talk, biological factors, behavioral, lifestyle, physical built-in environment, socioeconomic, cultural, healthcare system, access to treatment. So there is a whole shebang of factors that affects individuals over their life course from childhood to adulthood. And these factors also, there's plenty of evidence that they leave a mark on the biology, by epigenetic modifications that drive altered gene expression that leads to tumor initiation, progression, and that's a vicious cycle. So the challenge is pretty overwhelming, but there's a French philosopher who has said that there's no problem that can withstand the assault of sustained thinking, and problems definitely turn into opportunities when the right people come together. So I'm marching forward with a team of social scientists, public health experts, clinicians, biostatisticians, bioinformaticians to do as much as I can to find these answers. I have a few more slides. So harnessing the power of ICARD and transcending these disciplinary bounds, we have been able to publish a lot of other multi-institutional studies using multiple cohorts. Some of them are AI based, like this one and this one as well. So these are some of the questions that we are asking now. Is loss of AR associated with African ancestry? Is Wnt beta-catenin a therapeutic target? Does AR confer chemoresistance? And do PTMs play a role in disparity? Does centrosome amplification lie aggressive breast cancer in Black women? So so this is some more focused on the neighborhood disadvantage. Does living in a disadvantaged neighborhood impact treatment outcomes? So this is one study that's going on in the city of Atlanta. So these are some of my other community-based projects. So I'll just skip all of this because this is the other side of my lab, which is looking at intratumor heterogeneity using centrosome amplification and cell cycle traverse rate. Both of these are IP protected technologies. I can share offline if somebody is interested. And there are some AI-based studies that we are looking at. And of course,
please wind up now.
Yes, all of this is possible because of these multiple platforms. And here's my lab. I only talk. These are the people who do the work. And thank you so very much. I'll take any questions that you have.